It's okay. We haven't missed anything. We're just starting now. There you go. So. All right, let's compare three things. Now we're going to talk about translation. But before we start with translation, we want to talk about three things. Okay? Three things. Compare them. Genes. Whoa. Do any of you like those mutated jeans that have those holes in them? You know the jeans that people wear? They have a lot of holes? Yeah? I call them mutated jeans, right? They're like they're stylish.
Not anymore.
Really?
Well, for the guys, not anymore.
Only for girls? Yeah, girls can get away with anything. Guys can't. Girls can wear guys' clothes, no problem. We wear girls' clothes, we're in trouble. I have to be shifted to San Francisco, right? Okay. Genes. Now, genes, in terms of the length of sequence, varies, right? It's variable. Codon is only 3 nucleotide in length. Only 3 nucleotide in length. Anticodon? 3 nucleotide in length. Very good, very good. Gene is found on what? DNA. Okay. Although it's also transcribed in the mRNA, you can call it that, right? Codon? mRNA is much better than RNA. Yeah, I have to give her extra points for that. mRNA only, not on ribosomal RNA, right? Okay, how about anticodon? tRNA. Very good, very good. There you go. All right. Genes still have introns and exons in them. Codons and anticodons don't. Right? Genes have in their sequence some regions that are coding, some not coding, which means genes do have introns and exons, whereas these guys don't have. Okay. At the end of the day, gene is a sequence of DNA of variable length, depending on the gene. Codon is a sequence of mRNA, only 3 nucleotide in length. Anticodon is a sequence of RNA, only, what RNA? tRNA, and 3 nucleotide in length. One more thing, anticodon is complementary of codon. Anticodon is complementary of codon. Okay, it's complementary of codon. The first nucleotide must be complementary. The first nucleotide, usually the first two. The first two nucleotides are usually must be complementary. The third could vary. For codon and anticodon, the first two nucleotides of codon and anticodon must be matching, must be complementary. The third nucleotide varies. It's okay. Okay. Now, for example, not necessarily mutation. I'll tell you. In mutation, I'll discuss it more, but let me tell you this. In location, yes. The third nucleotide of anticodon is always variable. It doesn't matter if it's matching or not matching. The first two, it must be sequence. Let me give you the sequence of, let's say, a codon. What would be the first codon in mRNA? What has to be the first one? AUG. Right after that, as soon as you reach AUG in the mature mRNA, because each codon is 3 nucleotide, right? And then after you choose 3 nucleotide at a time, this is another codon. This is another codon. And this is another codon. And here, why am I choosing? I choose different color for this. This is red. This is another codon. Another codon. And then you have one more codon. If there's extra beyond that, that's okay. But let's say there's another codon. See, as soon as you find AUG, you start counting 3 nucleotides at a time. That becomes 1 codon. And what does codon code for? A single amino acid. A single amino acid. AUG, you should memorize it, always codes for methionine. Okay? AUG, you should memorize it, always codes for methionine. Which means usually in protein synthesis, the first amino acid is always methionine. Okay. Now, and think of start codon AUG. Usually, high school starts in August. AUG, August, start codon, if you can remember it. Okay. Now, you don't have to memorize the rest of them. I think CC stands for cysteine, I think, but I'm not sure. I think GG stands for glycine, but again, I'm not sure. You don't have to memorize because that will be given to you. Genetic code will be given to you.
Do we have to memorize it?
But we don't have to.
No,
Only the
it'll be
start.
given to you. I'll tell you what to memorize. GGA probably also stands for glycine, or let's say glutamate. I don't know. AAA I think codes for, I don't know, lysine I think. UU I think codes for phenylalanine, but again you don't have to memorize. Stop codon you have to memorize. What you have to memorize, here you go, I'll do this, check mark. AUG, you must memorize. Stop codon, you must memorize. Start and stop codons, you have to memorize. There's only one start codon, which is easy, August. Memorize that. And it codes for amino acid called methionine. Stop codons, there's 3 of them. I heard somebody saying UGA, UAA, or UAA, not UUA. UAA, UAG. Very good. This you have to memorize. So total of 4 codons you have to memorize. You only have to memorize 4 codons out of 64. 60 codons will be given to you on a sheet of paper on the exam as well. And you can use them to make your amino acid sequence. That's called genetic code. Genetic code is this codon sequence. Okay, now, how many nucleotides are there in the RNA? How many possible types of nucleotide do you have in RNA? 4. So, how many ways can you combine 4s into a nucleotide of 3? So that means 4, choose 3, if you did calculation in high school. Permutation combination, right? So this will 4, choose 3. I don't know if that's the symbol. Mathematics expert?
It has to be P, actually.
It's P? Okay, permutation, there you go. So you don't have to worry about this. I'm just trying to remember something. So this is 64 possibilities, the way you can combine 3 nucleotides at a time. And out of 64, 4 are the only one you have to memorize. You only have to memorize how many? 4. The other 60, all 64 will be given to you regardless. But 4, it's a good idea to memorize. AUG and stop codons. Now, there's a mnemonic for it. You go away. You are away. You are gone. It will help you to remember it, right? So protein synthesis, you tell the ribosomes, you go away, we're done. You are gone for sure. You, what else is? You are away. Don't worry about it. Don't make no more proteins, I guess. Okay. So memorize these. The rest will be, all of them will be given to you, all 64. But sometimes AUG and stop codons might not be given to you. You have to be able to recognize it. Okay? So that's one thing. Now, second thing is, how many amino acids do we have that participate in protein synthesis? 20. We have 20 amino acids, correct? Amino acids. How many codons do we have in total? 64. 3 of them are stop codon. Stop codons don't code for anything. They don't code for amino acid. Stop codons don't code for amino acid. Okay? Now you have how many codons left? 61. 61 codons will be coding for 20 amino acid. That means what? There are certain amino acids that will have more than 1 codon. There are certain amino acids that will have more than 1 codon. For example, glycine I think has 3. And I think phenylalanine has 4. Some will have 1, some will have 3, some will have 2. So there will be some amino acid that they will have multiple codons. Okay? And a term for this that is used is the genetic code is degenerate. Degenerate, meaning that there's some codons that'll code for a single amino acid. It's not one amino acid, one codon. Look, we talked about one gene, one protein, but there could be multiple of codon, maximum of 4 for one amino acid. Okay, because we have extra codons, we have limited amino acid. Make sense? But the genetic code will be given to you. It should be in your books too, if you look at that picture. Now, always the first 2 nucleotide of a codon will determine the amino acid. The first 2. It's the third one that's degenerate. I'll use X for that. The third one. The third one. For example, if you have UU, if all UUs stands for phenylalanine, then it doesn't matter if it's UUA, UUG, UUU, UUC. All 4 of them will code for what? Phenylalanine. So any codon that codes for amino acid is based on the first 2. The first 2 they cannot switch. If you change the first 2, that will lead to mutation. The last 2, depending how many amino acids would have that. So the third sequence or the third nucleotide is degenerate. It's called wobble. Okay, that means you can switch them, but the sequence of amino acids will not change. Okay, because all 4 of them can code for the same amino acid. No big deal. But if as soon as you make this UA, it's no longer the same thing. It could be UU, UAA, UAG. These are stop codons, right? It's a problem. Okay, so that's one thing. Now let's go. Sometime when we discuss mutation, mutation is any change in the sequence of DNA or in the sequence of gene or sequence of codon. So that means mutation can be anything there's a change in the sequence of DNA nucleotides or mRNA nucleotide or protein amino acid sequence. If you change the sequence of DNA or RNA such that the sequence of amino acid don't change, it's not a mutation, it's a silent mutation. It will not cause any problems. Because it's the product that's important. It's the product. Activity in your body is because of the product. Enzyme, product, right? Protein structure, product. So if the sequence changes such that the enzyme or the protein changes, it's non-functional, it's a mutation. But if the sequence of DNA changes and the sequence of mRNA changes, but the sequence of amino acid don't change, nothing happens. It is still a mutation, but it's silent mutation. Okay, it would have no side effects. It will not have the disease or anything like that. I'll give you some examples later on, okay? But let's start with protein synthesis, now translation. So once we have this mature mRNA, then the next thing is the large subunit of ribosomes will come. So first, who brings amino acid to the site of translation? tRNA. So we will have a lot of tRNAs. I'll just write T's with a loop. Now these tRNAs are just floating around.
This is translation right here.
Now we're talking about translation, yeah. These tRNAs are floating around. So the anticodon on this one is, let's say, G, G, G. Here is C, C, C. U, U, U. A, A, A. If you find anything abnormal, let me know, okay? So I'm going to try to do things so that, see if you can, okay? Okay. U, G, A. Or A, C, U. Okay. U, A, C. Okay, let's see. Let's make one more. A, T, G. let's make one more. Okay, let's make this one G. Whoa. G A. And then let's make last one C U. Okay. I'm just going to randomly assign amino acid because I don't know exactly what each sequence stands for. You'll be given genetic code again. We don't have to do that. Let's say this one carries amino acid known as glycine. Let's say this one carries amino acid known as mistake. Let's say this one carries amino acid known as arginine. And let's say this one is glutamate. Okay, let's say this one carries methionine. Let's say this one carries cysteine. Let's say this one carries phenylalanine. Let's say this one carries alanine. Let's say this one carries, what else is left? Valine. And this one will carry,
Stop quoting.
which one?
Stop.
Stop amino acid. Okay. Okay, I mean, you tell me. If there's mistakes, there's mistakes. Such thing don't exist, you let me know.
Maybe let you know now, or?
No, when I'm doing it. Yeah. Let me finish this. No, okay. Now, these guys are just simply found randomly. You know, they just swim around. They just run around randomly. Okay. Whatever codon and anticodon matches, only that tRNA will come and attach to that codon. This is very specific in terms of sequencing. The anticodon and codon must match. Otherwise, the tRNA cannot even come in. And how would you know what sequence of amino acid you have to attach? Right? Okay, so which of them don't belong here? Which tRNAs? Tell me the amino acid that's attached on top.
Valine?
Which one?
Valine?
Valine. No. Which one is invalid? Such tRNA don't exist.
Mistake, ADG.
Mistake, yeah. Because tRNA cannot have a T, right? And stop codon doesn't have tRNA.
No, that's not a stop codon. The other ACU is a stop.
ACU, where is that?
No.
UG is a stop codon on the mRNA.
The anticodon tRNA. Remember, it has to be anticodon. There's no tRNA for UGA. That's correct. So there's no such thing. There's no such thing. Okay. So this, let's erase and call it different amino acid. Just call it alanine or valine. There you go. Okay. Now, so the first thing what happens is the tRNA that corresponds to AUG should come and attach to it. What's that tRNA? Methionine. What color is that? Green. What if this guy comes first, glycine? Do you think it will come? It's random. It still comes, but it doesn't hold. It falls off. It moves on because there's no base pairing. Okay, so this one will not be able to. And they all come randomly. Whatever comes in contact first will come in contact. Let's say methionine comes in contact with this. It will be attached. It will stay here. The next thing, so this is the first step. Second thing is the small subunit of ribosomes. The small subunit of ribosome will now come and attach.
So AUG binds first.
tRNA binds first. And technically, the time frame is so short. Yeah. But they say tRNA binds first. Okay? But it's so quick that you can see spontaneously both attaching at the same time. Okay. Now, so that's one thing. Once the small subunit, let me put it here. What is the small subunit of ribosomes? What's the name for that? In eukaryotic cells. 40S. Okay, so we'll come and attach. Now, small subunit doesn't really have, in terms of peptidase activity, it only has stability. It only adds stability to this. The next thing that should come and attach is known as the large subunit. The large subunit, I'm running out of room. Can I make it somehow small? I think I can make this smaller. Where is those? Yeah, this used to, maybe this one. Let's see. Nope. There used to be a way that I could make it smaller. See, this is,
Isn't it cropping one? No, there's
no, this,
shrink the size.
I'll just do the whole thing. Maybe this is, they restricted it. There you go, maybe this will do it. It used to be much smoother, now they did make it difficult. Okay. So let's say the large subunit next, the large subunit will come and attach, but the large subunit has 3 areas in them. So this is the large subunit of ribosomes, 60S, right? 60S. The 60S ribosome has 3 locations. 3 locations, or you can think of it 3 garages, or 3 stops. A is the acceptor. So A stands for acceptor. I'll just write it and then I'll erase it. A is the acceptor. P, peptidase. E, exit. It has 3, in this case you can call them windows, but 3 locations, 3 areas. That means it can have 3 tRNAs at the same time. All tRNA have to come through here first. They have to be accepted first. The only that will be accepted is whatever is corresponding to the appropriate codon. And then it's moved on to the peptide location or peptidase activity and then exiting. Let me show you how that will work. So as soon as this binds, then the large subunit can come and attach. Even the large subunit will start with what? You know what? Let me do this. Erase this. I'll make it on the side. Oh. I will make it on the side. Let's make the large subunit right here. I want to make it so that it fits 3 at a time. They're tall. 1, 2, 3. I'm just drawing it. You don't draw it yet. Okay, I'm just drawing it. Just to give myself some. There you go. And you know the errors, right? Do I have to label that? Let me label too. So here is A site, and this is P site, and this is E site. Very good. So let's say this large ribosome is somewhere here hiding. As soon as tRNA and small subunit attaches, then the large subunit will come and attach. Of course, it will attach here first with the A site. Right? When it does so, and then it will recognize this as a start code, and it will move on 1 step to next. Once it moves on next, now what's the next tRNA? GGG, which carries glutamate. Okay? And glutamate will come and attach here. Okay? Glutamate will come and attach here. And I'm going to copy this so that way it makes it easier for me. Let's copy this. Uh-oh. How do you copy it? Can't copy? I guess not. So let's say glutamate comes, next one, and assume that this will come here and attach, because this will be now in the what side? The A side. Limitation of this technology. If you watch a movie, maybe that will be better, but let's see. Okay, there you go. The only complementary to CCC is GGG. So obviously that's the only tRNA that can come here. Now you see that the 2 amino acids are brought close together. The 2 amino acids are brought very close together. Methionine is brought close to glutamate. When they're close enough, you can form a bond. And the only site, or the only ribosomes, first of all, that has the ability to make peptide bond is what? Large subunit, 60S. Number one, write it down. Only 60S can make peptide bonds. Only 60S can make peptide bonds. The 60S of ribosome in humans or in eukaryotic cells. Okay, 60S. And within this molecule or this ribosome 60S, only the P site has the ability to make peptide bonds. And this means there must be an enzyme within the P site. And that enzyme is known as transpeptidase. Trans means combining two things. Peptidase, peptide bonds, or peptides. Transpeptidase. Only P has a transpeptidase activity. There are some antibiotics in bacteria that will inhibit transpeptidase activity, not in humans, and that will lead to killing of bacteria. Because bacterial ribosomes are different. So, okay, when this happens, next thing is this will move. It will scan and it will move forward. Now the A site is empty again. When it's empty, the next tRNA can come. And whatever the next one is should be CCC, right? There you go. This comes. Which means already there was a bond formed. So let's assume that we went through this already. There was a bond formed between these two. They were attached. Now you want to make attachment here when cysteine comes here. Okay? And then when methionine tRNA goes to the E site, the bond between the tRNA and methionine is broken. And this tRNA will go empty-handed, go get another methionine. So at the end of the day, you're just simply attaching one amino acid. You're simply attaching amino. Whoa. Where is it? Amino acid, amino acid, amino acid. You keep adding. You keep adding amino acid. The addition of amino acid is one at a time to this growing chain of amino acid or peptide. And this addition is only done in the P site of large subunit of ribosomes. Okay? Overall, now nobody cares for this process. Nobody's going to say you have to go through this entire process to remember, as long as you know the function of each thing. And this would be more important where? In bacteriology. Not necessarily in eukaryotic cells. But it's a good idea to know the process overall, how you make proteins. Yeah.
The 40S you said just attaches to the start codon.
No, it keeps, the two of them will scan together.
They scan together.
Yeah.
So the 40S will just scan wherever the 60.
Without 40S, 60S cannot attach.
Oh, 40S is
Yeah. Okay. And they'll both keep scanning the same way. It's almost like you have a tape and you have two things that go like this. Okay, and it keeps reading the code one at a time, one codon at a time, one codon at a time. And you keep adding amino acid in that specific sequence. Now you know why DNA is an instruction or a code. The sequence of those codon matters. You have 20 amino acids. The way you combine them, it's almost like lottery. You know, if you have 20 numbers to choose from, you know, and you can combine them in any way, in any length, it's impossible to have limits on that. That's why you have so many varieties of proteins or products. Because it's all depending on what? On sequence. Yeah.
Does the 40S stay attached to the start codon?
No.
No.
40S is needed for stability of large subunit. So both of them will move together.
So it's going to go like
Yeah,
this.
I just don't have, you know, enough technology to move them both at the same time. But they'll both move at the same time.
Okay, so like as the 40S, first the 40S will start at the start codon, so will the 60S, but the 60S starts at the top.
Yeah, when they're both attached, they keep moving together like this. Okay? As a unit. Yeah.
But one is at the bottom and one is at the top.
Yeah, sure.
Those are proteins, right?
Ribosomes are made of proteins and RNA.
I don't know the 40S.
Yeah, 40 and 60S are a combination of RNA and protein. They're a combination of protein and RNA. But yet they're involved in protein synthesis themselves. It's like making metal in a metal company, right? The machines are made of metal or iron, and you're making more iron, so same thing.
Is it rRNA?
RNA. Ribosomal RNA, of course,
Yeah.
and proteins combined together.
Correct.
Yeah. If you remember that we talked about this, the large subunit has how many? 33 proteins combined with ribosomal RNA, 5.8, 5, and 18. That's in your PowerPoints. You should go over those things. Okay.
The unit, the small and the large unit, they are promoting the tRNA to come, or what they do?
Okay, so let's talk about what is the purpose of the small subunit. The purpose of 40S is only to give stability or platform for 60S to attach. If 60S attach on one side, it can fall off, but if it's attached on both sides like this, it's much stronger. So the purpose of 40S is simply to give a platform for tRNA not to fall off, but for especially for 60S. Okay. Now, empty tRNA cannot stay there. Empty tRNA without amino acid will not be able to stay within the ribosomes. It's kicked out. And the only place you will have empty tRNA is in the E site. So when tRNA and amino acid attached to it reaches the E site, then the amino acid is cleaved, detached from tRNA, and then tRNA is kicked out. A site can only accept and occupy tRNA. The A site, so there are restrictions, of course. The A site can only allow a tRNA that has a cargo attached to it. You know, like if you have a docking for a ship or for trucks, whatever, if you don't bring the product, what are you doing here, right? So the purpose of tRNA is to bring amino acid where? Into the ribosomes. The A sites allows that to come in. It will only allow and occupy tRNA. A tRNA that has amino acid attached to it. It would not allow a tRNA that has no amino acid, even if it matches, the codon and anticodon matches. It won't allow. So once you bring it to the A site, and then that's transferred to the P site. In the P site, the two amino acids are attached. The peptide bond is formed where? In the P site. The transpeptidase activities only in the P site. Okay. The cleaving activity is only in the exit site, which is the bond between tRNA and amino acid is broken.
And what broke this bond?
The 60S itself. The 60S has intrinsic enzyme activity. It has intrinsic activity. In the P side, it has transpeptidase activity. In the E side, it has excision activity or breaking of the bond. But it's only between tRNA and amino acids, not between amino acids.
So the tRNA will remain without the amino acid?
It will not remain. It will be kicked out.
tRNA is kicked out.
Yeah.
All the tRNA is kicked out?
Mm-hmm.
So what's the purpose of bringing the tRNA if you are going to kick it out?
Transport.
Okay. Think about it this way. You have a taxi, and you know who brings amino acid to your factory. And the factory will combine amino acid to each other. Once you take the amino acid out of the taxi, what are you going to do with it? Tell them go back, pick more. That's the job of tRNA. tRNA is only for transport of amino acid. It's supposed to bring amino acid only to the ribosomes. Ribosome is the factory that makes protein. tRNA don't make proteins. Ribosome is the machine, the factory that manufactures or makes protein. Yeah. And protein is nothing more than combination of amino acids. The combination, how you're supposed to combine them in a specific sequence, that code is located on mRNA. You need mRNA to make a protein in a specific sequence. You need tRNA to bring amino acid to the factory, and the factory can combine it. So factory needs two things. One is the code. How should I combine these amino acids? Should methionine be first or cysteine? Because whatever you order, you have to have specific. But who is supposed to bring the product, or the amino acid is tRNA. Once the tRNA is empty, then you tell them go pick more. They keep circulating over and over. Yeah? Okay. So this is nothing more, this is very simple translation. It requires two ribosomes, small unit and large subunit. The large subunit is actually what makes peptide bonds. And specifically the P site. A site only accepts occupied, loaded tRNA. Maybe that's a better term. Okay. The A site of large subunit of ribosomes accepts only loaded tRNA. And the tRNA codon, anticodon should match the anticodon. So there's two criteria for the A site to accept tRNA. Number one, tRNA must be loaded. Number two, the anticodon must match the codon. Step one, step two. Once this is done, then the tRNA plus its amino acid is switched to the next site, which is P site. There, it could be attached in the growing chain of amino acids. Okay? Once the amino acid is attached, then the tRNA is separated from the amino acid. And it tells them, go. You go leave.
So there's going to be a mutation that occurs in the P site.
Yeah.
The one that's attaching.
I'll come to mutations. Good job. Yeah.
Are there 3 different tRNAs, or is it just one tRNA that moves from A, P to E?
No, it's the same tRNA.
It's the same one.
Whatever carries that amino acid. Let me do it different way. Yeah. Imagine this keeps moving. tRNA doesn't move. mRNA doesn't move. Yeah. This ribosome keeps moving on the mRNA. If there's a codon right here that is A, the only tRNA that can come here and attach is which one? Whatever has U. Okay? Let's say this tRNA comes and attaches here. Now, as soon as it attaches, this ribosome will move. And then this will come right here. Once it comes here and the peptide bond is formed, and then the ribosome will move one more step. And this will end up being here. And then once this is attached and it's here, the cleaving of the amino acid from tRNA is done. And then you have empty tRNA and it's kicked out. Because this will move out of the way. I should do it this way. Let's just for simplicity, think about this sequence. You have U, you have A, and you have C. Okay. Now what I have to do is I have to have tRNA is already attached just to make sense. Although that's not true, everything happens at the same time. Okay? Yeah. Yeah.
Okay, I had a question. So, does the same mRNA keep repeating that process, or is it a different mRNA each time?
Well, it depends what gene are you talking about. If you just want to make multiple copies of, for example, insulin, now you can have multiple copies of mRNA at the same time, all of them translated at the same time, you have multiple insulin protein, or you can have the same one repeated unless it's destroyed. So it depends on the time frame. If the polyadenylation tail is cleaved faster and there is no room for the second time, it won't happen. As long as mRNA stays stable in the cytoplasm, it will keep making and making. That's why it has to be cleaved eventually. Okay?
Will be the second one U.
Yeah. Oh, yeah. Yeah, Malcolm?
Is it true that there has to be a tRNA in the receptor site in order for tRNA to exit?
No. Okay, now, let's do this. So, here you go. Let's say this one has this amino acid attached, and this one has this amino acid attached, and this one has this amino acid attached. Okay, again, as I told you, tRNAs, when they come, the first one is okay, but the subsequent tRNA needs large subunit of ribosomes. Otherwise, they will not stay stable, they will fall off. Okay, so let's make a large subunit of ribosomes. Okay. Now let's say the large subunit of ribosome is over here. And in the P side is empty. Nothing is going to happen. It's going to scan. Go one more. Okay, so you have already one in the P side and then you have one in the A side. Okay. Now it will move so that you make a bond between these two amino acids. So now you have the E side and P side and the A side are all occupied. As I said, this technology is limited, so I can't. Here the bond is made between these two. Good? At the same time, once this bond is made first, then this one will be broken. So first the P side makes a bond between what? Between two amino acids. This is called peptide bond formation. Who does that? The P site. It has transpeptidase activity. When you do that, at the same time, you break a bond between the amino acid that is in the E site and the tRNA.
So make it between P and E or between P and A?
No, P and E.
P and E?
But it's the bonds are made here. Okay, it's just I have to, as I said, technology is limited. I have to put them in here. All the bonds are made where? In the P sites. In the P sites. In the P site. Let's just do this. I see where, because as I said, technology is limited. Let's put it this way. Hopefully this will help you. This tRNA is stretched. Hopefully this is much better.
Well, technically, that means the first one.
Now the bond is made. Where's the bond made? In the P site. Good? Okay. Now you have this. So, and then at the same time, as soon as this bond is made, this is broken. Now you have tRNA that is no longer attached to any amino acid in the E site. And let's say this thing has a stress, sort of like, you break it and it just becomes straight. It's stressful, right? So it's empty, and then the ribosome will move. Now again, I don't have room, so let's say it moves. Oh, I can do that. You guys should have told me I can do this. I don't have to keep trying. Yeah, let's move it. Let's move it. There you go. It moved to the next one. Now you see that there's no longer ribosomes occupying the tRNA. This tRNA will fall off and it will move on. It'll go pick up another amino acid. So now you have the peptide that is in the E site, and then again this will stretch here, and you will have one more bond formed in the P site. And then another tRNA can come here. Let's say the same tRNA goes. Luckily, let's say here is again the anticodon is, or the codon is AA. This tRNA, if it's fast enough, okay, if it's fast enough, although there's randomly more tRNA floating around, if it's fast enough to go and pick up an amino acid, another amino acid, it can come back. Yeah, it can come back.
And the tRNA will have much more amino acids all the time. The numbers will increase.
Only one tRNA, one amino acid. One tRNA can only bind one amino acid, but it can keep. It's almost like a.
You can keep going back to pick up amino
Yeah.
acids.
Yeah,
Like a
but
taxi
the
cab.
remaining amino acid, the bond that
Yeah.
formed with the amino acid,
Yeah.
it has like 2 amino acids.
Yeah, that's a chain. That's what you want to do. The end goal is to make a long chain of amino acids. That's what the protein is.
20 amino acid.
They are attached to the tRNA,
They're attached to the last tRNA.
to the last tRNA.
Yeah,
And it will keep increasing number.
exactly. So think about it now. If you keep increasing in number, and let's say there's 20 amino acids, keep attaching and attaching and attaching, the chain grows in this, and then whatever is growing, so this tRNA is attached to this amino acid at the same time, and this is attached to this. Once this bond is formed, and this tRNA is broken again, and this will leave. And then this will scan, another will come. You keep adding, one at a time, and you keep removing one tRNA at a time.
So eventually you don't have tRNA.
Let me find a movie for you.
I think the question is where's the tRNA getting the amino acid from once it's cleaved off?
The amino acid, cytoplasm is full of amino acids.
That's what the question
Yeah. So. like
You see, guys,
tRNA.
maybe I should do this quickly, okay? Good questions. Now I understand. You're made of cells, right? Let's say that my screen is entire cell. Or let's just make it, here you go. Just to make it simple. This is a cell. Everything occurs in the cell. When you eat food, you eat proteins, you eat carbs, and you eat trinucleotides and so forth, and what else? Lipids. So the only thing you can absorb from your GI is what, in terms of proteins? Amino acids. When this amino acid comes to the cell, it accumulates in the cell. If you need to use it, you will use it to make proteins. So amino acids are always available. Always within your cytoplasm. So they're always available. tRNA that's empty, and if, let's say this tRNA is for glycine, and it sees amino acid there, it will go attached to it. It's a random thing. They're just floating all over the place, like taxis. You find somebody, he says, hey, let's go in. Boom. Where you want to go? Here. Okay, let me go pick up someone else. There's never a shortage of people, unless you Uber, whatever, but you get the idea, right? Cooper. Okay.
Is Trans Properties also responsible for the cleaving of tRNA?
No, that's the E site. Yeah.
What do you say is responsible for this cleaving?
Yeah,
What does it
for
follow?
cleaving of the amino acid sequence or chain from what? From the tRNA.
But is there like a specific enzyme in that
I don't
site,
know.
or it just falls off?
I don't know the name. Yeah, I don't know if it's spontaneous or if it's enzymatic. I don't know. Okay. Good. At the end of the day, you will have a long chain of what? You will long chain of
Yeah, 'cause the bond for the
amino acids. Okay, you will have a linear chain of amino acids. True. And what do you call this? Peptide. Peptide. If it's very long, you could call it polypeptide. Polypeptide. If it's 2 amino acid long, what would you call it? Dipeptide. If it's 3, tri. If it's 4, tetra. And you get the point. How far are you going to go? We're lazy people. We don't want too many namings. If it's more than 10, you call it polypeptide. You can call it that too. Yeah. Okay, just for simplicity, you call it polypeptide. Usually what you're concerned with is dipeptide, tripeptide, and polypeptide. You always amino acid is a polypeptide. Okay, it's a polypeptide. Now this is still, a protein, sorry. This is called a nascent protein. Newly synthesized protein. This is newly synthesized protein. So now we're talking about the product of translation. The product of translation is what? Protein. Okay, but protein still needs processing. Now, you shouldn't be surprised. There are some amino acids that are water soluble. There are others that are lipid soluble. Some are negatively charged, some are positively charged. We have a whole section on amino acid metabolism, but I have to mention it here just for you to make sense, okay? Now, let's say we have red amino acid. They are positively charged. Let's say we have green that are negatively charged. And let's say we have these yellow, lipid soluble. Okay? Now the sequence of amino acid determines the property of a protein. If it's water soluble, lipid soluble. Good? Are we getting somewhere right now? At least? Okay. Now, so this is still a linear chain. Now, the final product is not a linear chain of amino acid. The protein will fold according to its properties. Do you think there's attraction between positive and negative, in this case green and red? Yeah. Do you think there's attraction between the lipid amino acids? Lipids attract to lipids. Charge, which is negative and positive, attract. Positive and positive repel. Negative and negative repel. That means if I have a long rope, when I throw it on the floor, I'm talking like Moses, haha, snake. But anyways, if I throw it, would it stay straight like a rod?
No.
Why does it fold? How about spaghetti? When you put it on a plate, why doesn't it just stay nice, straight, and clean? It will look good on a plate. Chef will have easy time, you know. Why does it fold? Some areas are softer. Some areas are rigid. So the folding occurs because of weakness and strength and so forth, the property of that. So when I take this linear polypeptide and just let it go in water after you synthesize, those areas that are attracted will come together. Those areas that will repel will go away from each other. At the end, the protein will have a certain shape based on its property. It will fold. Okay, it will fold. Now, protein function depends on its folding. Protein function depends on what? On its folding. So if you substitute amino acid in a sequence which is mostly hydrophobic, and you introduce amino acid that's hydrophilic, this protein will be now more water soluble. It wouldn't be able to stay in plasma membranes, for example. Let me give you an example of sickle cell anemia. There's single amino acid sequence difference, right? Does anybody know? Valine 4, glutamate. So the beta globin chain of hemoglobin should have, in the entire polypeptide, should have one amino acid that should be glutamate. And that glutamate is replaced, glutamate is water soluble, it's replaced with valine, which is lipid soluble. Now suddenly when this leads to a disease called sickle cell anemia, because the protein is misfolded now, the final product, because of one amino acid. Valine is hydrophobic, that means all those polypeptide chains that have valine in them, they stack with each other, especially upon deoxygenation. When these hemoglobin stack, they don't have the ability to carry oxygen anymore. And you start starving for oxygen, it causes disease and blah, blah, blah. So at the end of the day, number one, sequence is very, very important for a protein functionality. Mutation is when the shape of a protein is changed. Something similar, look. Heat can also change the shape of something. Egg is a protein. You fry it, it becomes something else. Okay, so protein functionality depends on temperature, pressure, and acidity, plus on its sequence. Okay. Environmental damages are what? Temperature variation, acidic variation, pressure variation, or direct trauma, physical trauma. Genetic is DNA code, mRNA code. Okay. At the end of the day, we are proteins. So it depends what damage is done to protein. Is genetic, you inherit it from parents because of code problems, or because of environmental problems. Okay. All right. Now, once this is done, so a protein, this is known as primary protein. Primary protein. This is known as primary protein. So I'll just, for me it's easier, let's make it a single line, primary protein. This primary protein, when it becomes helical, according to its properties, because it will have properties, negative will attract, positive will attract, will become helical, this is known as secondary. That's one shape, secondary. After it's made, the nascent, fresh protein made out of ribosomes is known as primary. As soon as it's released to water, it will start coiling. If it has that coiling property, there's only two shapes possible. One is coiling, alpha helix, or beta sheets. Beta sheets means they fold like paper. The protein will fold like paper. It's not coiled, it's just folding like paper. So this is known as beta sheet, and this is known as alpha helix. Beta sheet and alpha helix are secondary protein structures. Still not a final product. So primary protein, secondary protein, the final product we want is tertiary protein, and that is when this protein folds on itself randomly according to its properties. Okay, all of this will occur where? In the rough endoplasmic reticulum or in the cytosol. Okay, so this is know the primary structure of protein, secondary, tertiary. Now, quaternary structure is when you have multiple polypeptide attached to each other. Quaternary. We have primary, secondary, and tertiary. Okay, let's do this. Primary, linear sequence of amino acid, right? You okay with that? Secondary is alpha helix or beta sheet. Tertiary is when that alpha helix or beta sheet starts folding further, or starts folding. Linear, I'll put L, alpha or beta sheet formation, and folding is tertiary. Quaternary, now this was all for single polypeptide. Quaternary is when you have multiple polypeptide. After all of this has happened and they combine together. Let's say this is, look, this was a linear and this was alpha helix, and then the alpha helix folded on itself and it forms something like this. Now this is tertiary, right? If you take one tertiary and two tertiaries combined together, they're separate polypeptide, they're not a single polypeptide, then it's called quaternary structure. When you have more than one subunit of protein attached together, it's quaternary. Quaternary structure. Whoa. For example, hemoglobin is made of 4 subunits of polypeptides. One is alpha, another one is alpha, and then one is beta, another is beta. Each of these single polypeptide. Each of these guys are single polypeptides. That means they're all tertiary proteins. So this is tertiary, this is tertiary, this is tertiary, this is tertiary. For hemoglobin to be functional, all 4 of them have to attach together, make one unit. Then it's known as quaternary. When you have more than one protein fused together, when you have more than one protein fused together, or polypeptide chains fused together, it's quaternary.
What attachment?
Hmm?
What attachment?
A lot of things. Cysteine bonds, phosphorylation, we'll talk about it. Okay. All right. So this is protein synthesis. Now there's one more thing. Post-translation modification. It will take 1 minute. It will take a bit. Post-translation modification. Post-translation modification. That is, once you make a protein, you can add a phosphate group to it. It's called phosphorylation. Now you're changing the shape. You can add OH group to it. It's known as hydroxylation. Okay. You can add a sugar to it. Sugar is AR. That's known as glycosylation. You can add cysteine bonds. Okay. Disulfide bond. You can do many things. At the end of the day, once you make a protein, you can modify it further. For example, let's say you buy something, you're like, you know what? I'm going to frame it. Some say I want to frame it with gold. Some say I want to frame it with this, blah, blah, but you, according to your needs. That's post-translation modification. It's not necessarily done by ribosomes, but it's done by Golgi and who? RUF-ER. Excellent. And one extremely important post-translation modification is known as mannose.
6 or 7.
One less. It is 6.
Yeah.
Mannose 6-phosphate. When you add mannose 6-phosphate to a ribosomes, it is a signal for transport into lysosomes. As soon as you add this, this protein is targeted for lysosomes. Okay? In other cases, you can add an address on a protein. For example, if a protein is mostly membrane protein, you will have to add a lot of glycosylate, a lot of sugars, because they'll act as an antigen. We're going too much into specific, but just know that post-translation modification is when you change the shape of the protein, when you add something or you take something away. Okay, and it's only done in ribosomes, in RUF-ER and Golgi. Okay?
What's the last one?
Just adding an address by Golgi, like a stamp. It's a post office, right? So protein that are supposed to go to lysosomes, to mitochondria, or outside of the cell, or to cell membrane, you need to put a tag on it, because how would your cells know where to take this protein? And that's done by Golgi. Okay, let's take a break and I'll come back to this.